40 years ago, when the president of this academy was Carlos Chagas Jr. from Brazil, he asked me to help him in organizing a study week on non-specific immunity in cancer. At the end of the week, I was supposed to go and report to the Pope, which was Paul VI, Saint Paul VI, and I told him I never was at a meeting more exciting scientifically and more depressing medically. Now, 40 years went by, and we are having in a few weeks Nobel Prizes in medicine and in chemistry, and all are in immunotherapy. So there is a tremendous difference and progress in these 40 years. Now, I would like, do you hear me well? Yes. I would like, first of all, to make a couple of comments on the main topic of our deliberation, namely on emerging basic science towards solution for people's well-being. On the one hand, the drug we invented against multiple sclerosis, called Copaxone, improved the health of many hundreds of thousands of patients, and this helped their well-being and alleviated the worry of their families. At the same time, the drug, Copaxone, became the main export of the country and definitely improved its economy. Thus, what started as a totally basic research ended in significant improvement of health and a rise in the level of economy. What I would like to talk a little bit now is about curiosity-driven basic research, because this was not a translational study of looking for a drug for multiple sclerosis. Totally differently. We were working with synthetic polymers of amino acids, which were supposed models for proteins. Now, if there are models of proteins, for example, we prepared synthetic polymer resembling collagen, and we were very happy when antibodies to this polymer reacted with collagen, and antibodies to collagen reacted with this polymer. The same when in enzyme, we took lysozyme, we built just a little piece of it and attached it to a polymer, and then we got injected, we got antibodies reacting with the enzyme and vice versa. So it is in this spirit that one day I found that there is a protein called myelin basic protein. Basic meaning positively charged, and myelin because it's really in the brain and it's really on the nerves, the material that is stopping it from reacting one nerve with another and causing what ultimately is multiple sclerosis. So I learned that this material, this protein, injected in mice was causing them a disease which resembled the model for multiple sclerosis, allergic encephalomyelitis. So we decided immediately to prepare several polymers of amino acid in this spirit with a similar charge, much simpler, and we started injecting them in mice, and they never got sick. It took some time until we understood not only that they didn't get sick, but actually when injected in animals which had already allergic encephalomyelitis, they healed them. So at this moment, this became a translational research. It took 28 years from the moment of the idea to the moment of approval, the FDA. Now, in such time period, you can become an expert in multiple sclerosis. And this was after, but I want to drive this because it became so important. And it became so important in terms of royalties to the Weizmann Institute, and was all of this of curiosity-driven basic research. And I want to stress that I believe that actually most of the important discoveries are not when you try, President Nixon tried to repeat the success of putting a man on the moon by giving the same amount of money and solving cancer. But it doesn't go this way. So, in this spirit, this copolymer, which we used to call copolymer 1 or COP1, and then industry, as the disease is both immunological and neurological, they put two letters, AX, between COP and 1. The name Copaxone is to indicate that it helps. Now, close to 400,000 patients this time, but actually, over the years, it's probably close to 1 million treated just, which it's specific, it doesn't act as a general immunosuppressive modality. Recent work on an animal model of multiple sclerosis, experimental autoimmune encephalomyelitis, suggests that Copaxone not only prevents demyelination, but may even promote remyelination of the brain. Now, this is the first message I wanted to give, the importance of curiosity-driven brain research. For most individuals, after many years of research, publication in an impressive journal seems to be the end of the road. But this is not true, certainly not in this kind of research, the translation, because the next step, the development of fruits of research is most important. The translation of research into product is important both for the individual and for humanity as a whole. Now, while both achievements, health and economy, are significant, undoubtedly the greatest satisfaction for a scientist is the unexpected result in basic research. I don't see how much time I have. Yeah, no, I have plenty of time, but I can stop at any moment. I wanted just to give you a few examples about really, from early works on synthetic antigens, we were led to distinguish between immunogenicity, defined as the capacity of an antigen to provoke an immune response, and antigenic specificity, defined as the capacity of antibody to recognize the antigen. Thus, there might be antigens that react specifically with antibodies prepared against another immunogenic substance, but which are not able by themselves to trigger an immune response. Now, today, there are tens of biotech companies with the name of immunogen or immunogenicity, but it all started with our need for these expressions because we were working with this totally defined antigen. And I want to give this an example because the moment we have discovered what were synthetic antigen, it means that you can prepare anything you wish, and if you prepare the right way, you can have something which will lead to making antibodies. So the moment today, I think we can get antibodies to absolutely everything we need. Now, another example is, I said I wanted to mention 3 cases. So have a first synthetic antigen, that's general to defect antigen. Second is working with antigens that were chemically simple led us to discover the genetic control of the immune response by using different strains of inbred mice. And this was, again, an example which led to a whole field, the immunogenetics, which was very important. And the last example I'll give, after so many years of working with models of proteins, there was time to work with proteins themselves. So 6 years ago, I went to Chris Anfinsen, and there we showed that there is no need for additional genetic information to allow a polypeptide chain to fold into the desired protein's 3-dimensional architecture. This was the conclusion of opening the 4 disulfide bridges of pancreatic ribonuclease by oxidation, thus impairing its enzymatic activity, and then letting it reform on its own and regaining its enzymatic function. So this was very important, but as usual, until today there are hundreds and thousands of papers showing the details. The problem is not totally solved, but this was the first case that showed that you don't need additional genetic information. I think that's all I want to say. I dropped after 5-6 minutes, but essentially, I think the main message is that the curiosity research, basic research, is the one that leads to greatest discoveries and most important, and it takes a long time until you can accept it as a drug, because 28 years is also until the first statins were approved, exactly 28 years, when something is totally novel. Thank you.